Yeah, this part 3 is foodborne illness caused by virus. There are two viruses which are causing the foodborne illness. One is hepatitis A virus and the second one is norovirus virus. So before discussing these foodborne illness caused by these kinds of virus, I will explain you brief about the virus, introduction of the virus, origin of the virus, and the different sizes of the virus or shapes of the viruses. And another one is how the viruses are evolving into the new, emerging into the new mutant species, how the virus are mutating. So virus is a infectious particle. It's neither living thing nor non-living thing. So sometimes it exhibits living characteristics and sometimes it exhibits non-living characteristics. So virus is a intracellular obligate parasite. Virus is acellular. A virus can replicate within the host cell only, specific to the particular host. So virus is neither prokaryotic nor eukaryotic. Virus is nothing but a virus is a poison. It's a liquid poison virus. Virus can multiply, virus can replicate by using matured virus is called viron. Louis Pasteur first studied the rabies virus in the history, and the virus which can infect the bacteria is called bacteriophage. When the virus enters into the host cell, then it will take time to exhibit the symptoms. The time period is called incubation period, and the virus from the infected person to healthy person will transmit through the direct contact of the person or the person, infected person who contacted the objects by touching the healthy person, the same object will transmit the virus, or sometimes a virus can transfer through some mosquitoes, insects also. So those insects act as a vectors, and the virus won't harm to the vectors, won't harm to the insects. So some vectors act as a reservoirs. So the virus which are living inside the vector for many days, it acts as a reservoir in a latent period. And some hosts also, after entering the virus into the host cells, it will be dormant state, means in a latent period. Some virus, for example hepatitis A virus, many viruses are there. So once it will enters inside the host cell, it won't mature, it will become in a dormant state, and when sometimes the favorable conditions prevail, then the virus will replicate and will infect the tissue, then the symptoms will appear. Depends upon the genetic material, the structure of the virus. The structure of the virus is of two types, depends upon the genetic material: DNA virus and RNA virus. DNA virus is deoxyribonucleic acid and RNA ribonucleic acid virus. Most of the plant viruses are RNA virus, most of the animal viruses are DNA virus. Devolution or regressive hypothesis suggests that virus evolved from the free living cells, and progressive hypothesis suggests that virus originated from RNA or DNA molecules that escaped from the host cell. There are different hypotheses are there, origin of the virus. And in the genetic material, based on the genetic material and structure of the virus, viruses contains DNA virus or RNA virus, and the genetic material also in single stranded or double stranded, single stranded RNA or double stranded RNA, single stranded DNA or double stranded DNA, virus like that. And that depends upon the genetic material. Viruses are two types. Depends upon the shape, viruses are four types. And the size of the virus is ranging from 20 nanometers to 300 or 250 nanometer size. It's a very small virus. It's infectious particle. Virus is smaller than the bacteria. Then we will talk about the different types of virus. Based on the shape, viruses are classified into 4 groups. The one is filamentous. The second one is isometric, and third one is envelope, fourth one is head and tail. The first one is filamentous or helical virus, most of the plant virus. Then second one is isometric virus, isohedral type virus. For example, norovirus virus, hepatitis virus, all will come examples. Then the third one is envelope virus. Envelope virus means, for example, this COVID-19, which comes under the envelope virus. Envelope virus means, envelope virus is more susceptible for the resistance. Means we can easily control the enveloped virus compared with non-enveloped virus. This envelope is nothing but a lipid layer covering the protein coat of the virus. So virus, the genetic material is protected by the protein coat. The protein coat is called capsid, and this protein coat also the covering the envelope by the lipid layer. Like this COVID, most of the flu viruses having the lipid layers. So those are comes under enveloped viruses. And non-enveloped viruses means hepatitis virus or mumps virus or rabies virus are there. Non-enveloped virus. Most of the flu causing infections causing the viruses are enveloped viruses. So we can develop our immunity system and we can control those viruses. These isometric virus is different geometrical patterns, 5-fold or 3-fold, 2-fold, like that. It's isometric, different shapes of virus. And the third one is, we discussed in the enveloped virus. The fourth one is head and tail. Head and tail means bacteriophage virus, the virus which infects the bacteria. So the virus which contains head means the genetic material which is covered by the protein coat, like head shape, and the tail is it will attach the host cell. So for that only that. So attachments. And how the virus will replicate. So virus will replicate in 6 stages. The first one is attachment to the host cell, and the second one is penetration to the host cell. Then third one is uncoating the virus, and fourth one is replication. Fifth one is after replication assembling the replicated genes. Then sixth one is developing. After replicating, then assembling. Then sixth one is release virus to the outside of the cell. These are the 6 stages the viral replication occurs. So the virus replication in 2 types. One is lytic cycle and the second one is lysogenic cycle. The lytic cycle is very simplest method. So by lysis of the cell, lysis of the host cell, the virus will come out. Lysogenic cycle, it includes the phages, different phages. Most of the bacteriophage viruses contains lysogenic cycle, and some viruses which exhibits both lytic cycle as well as lysogenic cycle. What will happen, the first step, when the virus attaches to the host cell, it will penetrate the genetic material into the host cell, and once the genetic material enters into the host cell, the genetic material induces to the host cell genetic material, host cell nucleus, host cell organelles. It will utilize the host cell genetic material and it will replicate the same viral strains. It will replicate, and after the replication, then assembling, then come out. So before coming out, the cell will burst by the virus. Either the virus will burst out the cell or the cell will die, then the virus will come out and again attack the other host cells. So this is the way how the virus will replicate inside the host cell and again replicating and attack. Yeah. During replication, what will happen? So the virus, the genetic material like DNA or RNA virus is there. So it will incorporate itself into the host genetic material that induces to replicate the viral genome. So that is the process, and after replicating the viral genome, it will assemble and it will come out. There are about 5,000 strains of viruses are there, which are causing infections in humans, plants, animals, like that. So the virus, how the virus will mutate. For example, I will give you just example. In the year 2003, SARS, severe acute respiratory syndrome. So it's a kind of flu disease. Most of the virus, bats are the reservoirs. The virus which causes flu in bats, the virus which causes flu in animals, so due to the natural selection or unfortunately, so met together and changed their genetic material and transmitted to the human beings, mutated. So how the virus will mutate like that? So the similar kind will the genomic sequence. It's very easy to conduct the research on the COVID-19 and to release the vaccination, or otherwise to release the passive immunization. In different ways they can try to sort out the issues. So the genome sequence also, most of the virus contains maybe 5,000 genomes, maybe 5,000 genes, like that will be there, the genetic material genes. When we talking about this, the different viral strains which causes the flu in animals, which causes the flu in birds, met together and exchanging their genetic material, which will become more pandemic. The same thing happened to the COVID-19. So compare with earlier the SARS in the year 2003 and in the year 2013 also MERS, Middle East Respiratory Syndrome. So the virus mutated from the camels, bats, camels and humans that came. The recent virus also like that, mutated. The same flu virus. See, the virus which affected to us like COVID-19, so this virus won't impact on the bats, or this virus won't impact other organisms. It has mutated. So the sequence also they have revealed. So this is how the virus will mutate in every year, maybe in 5 years or in 10 years, depends upon the natural selections. So this COVID-19, and maybe after 5 years or 6 years, 7 years, again this COVID-19 will mutate. For example, one less harmful or no harmful virus and another no harmful virus, the similar strains met together and exchange their genetic material. Maybe the virus will come bit harmful virus or very less harmful virus, or one harmful virus and one no harmful virus, due to the natural selections or maybe the changing the genetic materials, they will mutate, may become more dangerous virus, or may become less or no danger, like that. So virus genetically mutate. It's natural selection, adaptation. So virus keep on mutating genetically, may become more pandemic, may become more vulnerable, may become more potential.